There is a fish swimming in the salt marshes of Southern California right now. It looks healthy. It schools normally. It eats. It grows. It reproduces. It also has thousands of parasitic cysts packed around its brain. And those cysts, quietly, without killing the fish, without disrupting its daily life in any visible way, have reprogrammed one very specific thing: how it moves when a bird is nearby. Not its whole behavior, just that, just enough to get itself eaten. This is not science fiction. This is happening in real time, in real water, to real animals. And the more closely biologists look at the natural world, the more they find it everywhere. Nature has a trick. It doesn't need to build a faster predator or a smarter prey. It just needs to rewrite the software of the animal it's already inside. And then sit back. Today, we're looking at 4 real cases of parasites that manipulate animals with a precision that, honestly, should not be biologically possible. Let's start with the fish. The California killifish is a small, unremarkable creature that lives in shallow coastal estuaries. Its parasite is called Euhoplorchus californiensis, a flatworm that begins its life cycle inside a horn snail. Larvae emerge from the snail, swim into open water, and penetrate the skin of any killifish they encounter. Then they migrate upward, into the brain. But here's what makes this different from most parasitic brain infections. The cysts don't damage the brain, they coat it. Researchers have found fish carrying hundreds, sometimes thousands of these cysts, arranged across the outer surface of the brain like tiles on a floor. The fish appears neurologically intact. It schools, it responds to stimuli, it functions. Except when a predatory bird is present. When a shorebird approaches, infected killifish begin performing what researchers call conspicuous behaviors. They dart, flash their silvery bellies toward the surface, swim in erratic, jerking spirals. Behaviors that are extraordinarily visible from above. Behaviors that a healthy, uninfected fish almost never displays. The result is not subtle. A 1996 study published in the journal Behavioral Ecology found that infected killifish were between 10 and 30 times more likely to be eaten by birds than uninfected ones. 10 to 30 times from a single parasite species. The parasite's goal, of course, is to end up inside the bird. That's where it reproduces. That's where its eggs are produced and released back into the water to begin the cycle again. The killifish is not a home, it's a transport vehicle. And the parasite has evolved, over millions of years, a neurochemical override for the exact behavior needed to get that vehicle delivered. Researchers later found that the infected fish showed measurable changes in serotonin and dopamine levels in the brain, the same neurotransmitters that regulate movement, arousal, and impulse control across virtually all vertebrate nervous systems. The parasite isn't randomly disrupting the brain. It is targeting specific chemical systems to produce a specific behavioral outcome. The fish never knows what's happening. It just swims until it doesn't. Our second case takes place in a forest in Europe, and it involves a snail whose eyes have been replaced with something that shouldn't exist. Leucochloridium paradoxum is a flatworm. Its final host is a bird, any of dozens of passerine species, from sparrows to crows. The problem is getting there. Birds don't eat flatworms. They eat caterpillars. So the parasite became a caterpillar. When the flatworm infects an amber snail, usually by way of bird droppings the snail has foraged on, it grows a structure called a brood sac inside the snail's eye stalks. These brood sacs are not just present, they are vivid. They pulsate. They are banded in bright greens, yellows, and browns. They throb rhythmically from within, expanding and contracting at the exact frequency of an insect larva moving inside a translucent casing. What the bird sees, from a branch above, is not a snail. It sees what appears to be a fat, juicy caterpillar, wriggling, exposed, easy. But the parasite does not stop at visual mimicry. It changes the snail's behavior entirely. A healthy amber snail is a creature of shadow. It forages at night. It retreats beneath leaves and into crevices when light increases. Every instinct it has is oriented toward not being seen. An infected snail does the opposite. Studies conducted in Białowieża National Park in Poland, published and peer-reviewed, found that infected snails climbed higher on vegetation, stayed in open, well-lit areas for significantly longer, moved more actively, and took more erratic, exposed paths. They became, in the most literal sense, animals that wanted to be found. Not because they wanted to, because something inside them had rewritten what they wanted. A bird lands, pecks off the eye stalk. The snail, remarkably, does not die. It can regrow the eye stalk. It can be reinfected. It can become a delivery mechanism for the parasite multiple times over the course of its life. The snail is not prey. It is infrastructure. The third parasite operates on a completely different scale, and its manipulation is not behavioral but architectural. The lancet liver fluke, Dicrocelium dendriticum, has a life cycle so improbable it reads like a design problem someone solved with a series of increasingly strange decisions. Its final home is the liver of a grazing mammal, a sheep, a cow, a deer. To get there from a snail, it needs to cross an impossible distance. It needs a land mammal to eat an ant. Grazing mammals don't eat ants on purpose. They eat grass, so the fluke puts the ant on the grass. After traveling from snail to ant, most of the fluke larvae distribute through the ant's body as they normally would in any parasitic infection. But one larva does something different. It travels directly to the ant's brain, the subesophageal ganglion, and installs itself there. It does not feed, it does not reproduce, it does not grow, it simply waits. And from that position, every evening, when temperatures drop, it sends a signal. The ant climbs. It walks to the very tip of a blade of grass. It clamps its mandibles down, and it stays there, completely motionless through the night, exactly where a grazing animal's mouth is most likely to sweep through in the morning. Here is the part that makes this particular manipulation remarkable, even among parasites. If the ant is not eaten by dawn, it goes back to its normal life. It rejoins the colony, it forages, it functions as a normal ant until the evening, when the cycle repeats. The controlling larva keeps the ant alive, keeps it functional, keeps it available, because a dead ant on the ground is useless. A living ant that climbs every night, for weeks, for months, until it finally ends up in the right mouth, that is a delivery system. The parasite has essentially created an alarm clock inside the ant's nervous system. Every evening at temperature drop, climb. Every morning, stand down. Repeat until consumed. The ant experiences this every day of its remaining life and has no mechanism to resist it, recognize it, or escape it. Our final creature does not manipulate behavior. It manipulates the body itself, reshaping its host into a form that will guarantee predation. Riberoia ondetre is a trematode that needs to complete its life cycle inside a fish-eating bird. Its intermediate host is a frog. The problem is straightforward. Birds are more likely to catch frogs that can't escape. So Riberoia doesn't bother reprogramming behavior, it reprograms development. When Riberoia larvae infect tadpoles at specific critical windows during limb development, they physically disrupt the cellular machinery that builds limbs. The result, documented extensively in Pacific tree frogs and other amphibian species across North America, is frogs born with severe malformations. Extra limbs growing from the wrong places. Limbs that are partially formed, rotated incorrectly, or entirely absent. Frogs that cannot jump properly, or cannot jump at all. A frog that cannot jump is a frog that cannot escape a bird. Field surveys have found that areas with high Riberoia prevalence show amphibian deformity rates as high as 90% in some populations. The bird eats the frog. The parasite ends up in the bird's gut. Its eggs are released into the water. A snail ingests them, and the process begins again. What Riberoia demonstrates is perhaps the most disturbing principle in this entire field. Behavioral manipulation isn't even necessary. If you can reach the body early enough, at the right developmental moment, you can simply build the outcome you need directly into the animal's physical form. 4 parasites. 4 completely different mechanisms. 1 shared logic. The host's body is not the parasite's enemy. It is the parasite's only resource. And evolution has had hundreds of millions of years to figure out exactly how to use it. There's a concept in evolutionary biology called the extended phenotype, first articulated by Richard Dawkins in 1982. The idea is that a gene's effects don't stop at the boundary of the organism that carries it. A parasite's genes can express themselves through another organism's body and behavior, just as surely as if those behaviors were the parasite's own. The ant climbing the grass blade is not displaying ant behavior, it is displaying fluke behavior. The killifish spiraling toward the surface is not displaying fish behavior, it is displaying worm behavior. The animal is still there, its neurons are still firing, its legs are still moving. But somewhere in the chain of cause and effect, somewhere between the parasite's chemistry and the animal's next action, the decision has been outsourced. The ant doesn't know it's climbing to die. The fish doesn't know it's advertising itself to birds. The snail doesn't know its eyes have become someone else's lure. And that, more than the grotesquerie, more than the precision, is what makes these systems genuinely unsettling. Not the manipulation, the invisibility of it. The host never gets the chance to refuse. The takeover is so complete, so seamlessly woven into the normal fabric of behavior that there is no moment of resistance, no alarm, no awareness of intrusion. There is only the behavior and the parasite moving quietly toward wherever it needs to go next.